Okay, thank you so much to the organizers for having me. It's my pleasure to talk to you a little bit about a female gynecologic cancer called ovarian cancer, which we study extensively in our lab. Ovarian cancer is the fifth leading gynecologic malignancy, and it's the most lethal. And although these numbers reflect the data from the United States, these numbers are the same worldwide. And that is because there is no early detection for this disease. Most women present with advanced metastatic peritoneal disease, for which there are very few personalized therapies that have been approved over the last 20 years. Essentially, all women are treated with frontline therapy of paclitaxel and a platinum agent, of which is currently there's a shortage. And then if you have a BRCA mutation, you may be eligible to receive PARP inhibitor therapy. So this tumor type requires a lot of study to understand its origin, the etiology, and new therapeutic modalities. We study one of the risk factors for ovarian cancer, ovulation. So every month, the extrusion of the oocyte from the ovary is actually a risk factor. And we know this because women who take birth control, oral contraceptives, or have multiple pregnancies are actually protected against developing ovarian cancer. Now, although we call this ovarian cancer, that is really an umbrella term. When we think about this in terms of mutational burden and different alterations at the genomic level and also at the transcriptomic and other levels of regulation in the cancer cell, we can actually see that high-grade serous subtype, which is the most common and most deadly type of ovarian cancer, does not arise in the ovary itself. It actually comes from the very tips of the fallopian tube, what we call the fimbriated ends, which touch or lay within proximity of the ovary. So it is a spatial problem. That intersectionality between the tips of the fallopian tube and the ovary is the place where we see malignant transformation, not uniformly along the fallopian tube, suggesting there are chemical gradients, specific short-acting molecules that are uniquely transformative at this interface. And this has been very challenging to study in animal models or humans because many of these secreted factors go within the belly, and they're not easy to understand what their concentrations would be, on top of ethical concerns that we would have digging into a peritoneal space to measure those concentrations over a menstrual cycle. So we can see, though, that the ovary is a contributing factor for disease aggressiveness. In other words, if these lesions remained in the fallopian tube, they're tiny microscopic in situ carcinoma. It is spread from the tube to the ovary, the initial primary metastatic site, that increases aggressiveness of these tumors. And we know that through things like transgenic animal models, where we remove the ovary and we actually see these animals live much longer and have less peritoneal spread. And also in studies done by my own laboratory, where we've seen that xenografting or allografting, depending on if it's syngeneic or not, that these cells actually form tumors much more aggressively within proximity to the ovary as compared to the same number of cells just in the peritoneal space. So many years ago, I'm embarrassed to admit how many, over a decade, we started working on modeling the female reproductive system using three-dimensional organotypic cultures, and we partnered with Draper Laboratories to create a perfusible technology that allowed for the interface between not only the ovary, which was derived from the mouse, but primary human tissues of the reproductive tract, including the fallopian tube, the uterus, the cervix, and also liver spheroids. We called this EVATAR, and what was really exceptional about this model was that it could recapitulate a 28-day menstrual cycle in terms of the mouse ovarian steroid and peptide hormones such as estrogen, progesterone, inhibin A, and inhibin B over a 28-day cycle, which is quite unique given that rodents normally menstruate every 4 days, or they don't menstruate, but they ovulate every 4 days, sorry. And so this was really interesting and phenomenological, but allowed us to create what was the equivalent of a human cycle in a microfluidic system. Since that time, we have worked alongside Draper Laboratories to change this technology for several reasons. One, to make it higher throughput. So we've now gone from being able to create kind of one interaction on a chip to, they have one platform called Predict 96, which is actually 96 individual microfluidic perfusible channels on a chip. That actually wasn't optimal for ovulating ovaries as well as the primary human fallopian tube tissue that we needed. So we worked with engineers at their company to engineer 30 individual cultures on the dish. So kind of like a 24-well plate, but 30. In this device, we actually see in the lid, there's a reservoir for the media, and there's also pumps that allows the media or the acceptor to go underneath or perfuse underneath the tissue which is being grown in the platform, and then the waste can be collected to study different steroid hormones or other products that are produced by the tissue or the interaction of the ovary with the fallopian tube. And since that time, we've gone on to also develop several insert technologies that allow us to do things like put an 8-micron pore size on a tissue scaffold so that we can fluorescently tag cancer cells and watch them through confocal microscopy migrating towards the gradients that are produced by the ovary over the cycle, as well as co-culture together cells that we can derive that are models of ovarian cancer from fallopian tube, preneoplastic lesions, cancer models, etc., together with an ovulating ovary. And so all of these insert technologies have really allowed us to push the forefront of this away from primary human tissue into cancer cells and cancer cell models as well. So I'll tell you one little story to start with about a protein called Versican that we have been working on that's a chemoattractant of ovarian cancer cells derived from the fallopian tube to the ovulating ovary. And so again, like we were able to show on previous microfluidic platforms, murine ovaries exposed to follicle-stimulating hormone and the luteinizing hormone mimetic chorionic gonadotropin ovulate on the platform and produce hormones similar to the female reproductive hormones. And what we were able to show is that when we collected media and did proteomics, and we could find factors in the media that were actually caused migration of both normal fallopian tube cells as well as cancer cell lines. I'm not showing you all of the data, but we did molecular weight fractionation to show that it was more than 3 kilodaltons. We then used heat and proteinase K to demonstrate that this was not a small molecule, but rather a protein. We did further size fractionation to identify that it was a protein between 50 and 100 kilodaltons. Again, more proteomics to show that the proteins most likely enriched in this had EGF-like domains, a small molecule screen to show that the EGF receptor was necessary, confirmed the EGF receptor was expressed in both a human and murine fallopian tube line that we used to study this disease. But of course, it wasn't that easy. It was not EGF itself, although that would drive the invasion. Instead, it was an unknown protein with EGF-like domains. This protein in this conditioned medium could be blocked by EGFR inhibitors in both a mouse and a human cell line model. So what was it? It ended up that when we looked at the 50 to 100 kilodalton fraction and then did proteomics specifically identifying proteins with an EGF-like fold, we identified Versican as our most likely hit after purchasing recombinant protein and doing other bioinformatic approaches. We were also interested in Versican because previously we had shown in high-grade serous peritoneal spread that it was important in spheroid formation in ascites. So again, our ovary conditioned medium produced Versican, and our ovaries ovulating on the platform are viable. We can see follicles growing within them, and we can actually see ovulation where the oocyte is being extruded into the platform. We can confirm all of this by using ELISAs to study the estradiol peak, as well as the Versican production. Again, connecting the idea that ovulation is a risk factor for ovarian cancer, suppression is protective. Versican peaks as part of ovulation. Therefore, this may be a contributing factor between ovulation driving preneoplastic lesions through a secreted protein tropic factor, Versican, to the ovary. We purchased recombinant Versican and did dose-dependent experiments in mouse and human cell lines and could show in a simple wound closure assay that this would drive the migration of these cells, and we could inhibit this using both a pan EGFR inhibitor as well as erlotinib in both a mouse and human cancer fallopian tube line. And then we used invasion assays using matrigel-coated Boyden chambers, again, to show that Versican was capable of enhancing invasion, both a mouse and human line, and that these were blocked by EGFR antagonists. Then, of course, we repurpose our ovary model now from a hormone-producing organ that contributes to disease formation from ovulation to now a metastatic site. So we're using fluorescently tagged fallopian tube lines and studying whether or not they adhere and invade into these 3D ovaries to understand if Versican is contributing to not only the chemo attraction to the ovary, but also the adhesion and invasion into it. So here we're again adding Versacan to the media, and we can see that this in fact does drive adhesion of fallopian tube cells to the ovary. Again, this can be blocked partially, at least in the human model, with the EGF inhibitor. So the first story I've told you is one where the microfluidic platform is able to show us what the ovary is producing in real time, or as we collect media and measure it in ELISA. We can then look through either proteomics in an unbiased approach or through specific ELISAs or other technologies for specific proteins, in this case Versacan. We can see that it correlated with the process of ovulation, and we identified that through EGF, in data I didn't have time to show you, that it activates phosphorylation that drives the migration of both normal cells as well as transformed fallopian tube models to the ovary, where they then adhere into this ovary as part of this initial metastatic spread. So we also have been using this platform to study many other reproductive issues. And one of the most common challenges that many women will face, about 1 in 5 women of reproductive age will have what's called polycystic ovarian syndrome. And one of the most common features of this syndrome is a high production or hyperandrogenemia. And what's really kind of interesting about this is that these women actually ovulate less. So they tend to actually have ovulation suppression. And if you were listening to anything I said, then you should be thinking automatically that they're protected from developing fallopian tube cancer, because I told you things that block ovulation are protective. Yet this group of patients doesn't seem to have protective effects against polycystic ovary syndrome, and while the data are very mixed, some meta-analyses have suggested they actually have an increased risk, specifically of what we call endometrioid subtype of ovarian cancer, which my and other labs have also shown the fallopian tube can be an origin cell type 4. So we were very interested in not only trying to develop model systems of PCOS, which Dr. Kim talked about earlier today, but trying to understand what the role was of testosterone on fallopian tube biology. So we used the microfluidic system in order to have 2 different scenarios where we had physiologic levels of testosterone that we would normally see in cycling women versus the roughly 2-fold higher levels that you'd see in patients that have polycystic ovary syndrome. And what was interesting, using RNA sequencing analysis, again, these are all primary human fallopian tube tissues grown in the microfluidic device, exposed to either low or high levels of androgen, is that we could see that there was a remodeling of the machinery that's responsible for making cilia. And cilia are important in reproductive biology for 2 main factors. One, they encourage oocyte rolling, so when the egg is extruded from the ovary, it actually needs to roll into the fallopian tube, and also sperm capacitation. So when the sperm are moving up the fallopian tube to hopefully meet the egg for fertilization, there's also binding, which facilitates maturation and capacitation of those sperm. So in both cases, if cilia are impaired, this could have an unintended negative consequence on reproductive function that Julie showed you earlier, where we co-culture human tissue with an ovary now triggered to produce high androgen on its own through exposure of LH. We see an even stronger repression of cilia beating in the microfluidic platform. We also looked at the human tissue in terms of proliferation, and we saw that in the microfluidic we actually had very high pCNA staining or proliferating staining in response to androgens, and this has not been appreciated before, because previously when studies have looked at testosterone on ovarian cancer, they have not studied the fallopian tube as the originating cell type. So this was not understood before. And yesterday, my research associate, Professor Angela Russo, so sorry if you didn't see it yesterday, but there was a poster where she took that idea, carried it further, and actually revealed through RNA sequencing in primary human tissue that there's an expansion of the cancer stem cell niche in response to testosterone. And in untargeted metabolomics data that we acquired through imaging mass spectrometry, we could see that fallopian tube tumor cells grown in proximity to the ovary actually trigger the ovary to produce more testosterone. So all of this suggests, again, that using these systems can really identify new biology and start to allow us to question or maybe repurpose other personalized medicines that are in our toolbox, like androgen antagonists, which have shown some success in early trials, but were never used in targeted populations of patients that actually expressed androgen receptor. And last but not least, I'd like to talk about an early detection project. So together with Dr. Godwin's lab at the at Kansas University Medical Center, we've been looking at extracellular vesicles as a possible biomarker. I told you there's no early detection for ovarian cancer, but what his lab has been able to show is an advanced disease, you can separate patients with and without ovarian cancer looking at extracellular vesicle cargo. However, we have no idea what the normal fallopian tube from human express in those cargo, so we don't know how they're shifting as disease accumulates, so we can't use those to predict early disease yet. So using the microfluidic platform, we were able to show that we can actually capture extracellular vesicles in high enough quantity, isolate them, and do proteomics on them, identify that, first of all, there's significant patient variability. When you unblind the cause or the reason these women had gynecologic surgery and donated their tissue, we can actually see that patient number one had a completely different reason for donating tissue. So these EVs may be useful not only for ovarian cancer detection, but other things. And when we look in terms of species similarity, many of these EV cargo have known reproductive function in other species. We can extend that to some human biology, as well as use it to predict which proteins may be cargo of these early lesions. And many of these cargo already in these EVs have known reproductive and cancer function in high-grade serous carcinoma. So I've shown you that the ovary can ovulate and is a risk factor. You can study mechanisms on the platform. Versican is one of these targets. Testosterone is another. And these microfluidic platforms are very useful, possibly for identifying new biomarkers. So thank you very much, specifically to Angela and Dee Dee, for much of the work that they did, as well as my collaborators, and I'm happy to answer your questions.
Thanks, Joanna, for this great talk. Questions?
Joanna, thank you very much. Regarding this last issue about the EVs and the comparison, for you to use them as biomarkers, you will have to isolate them from blood samples.
Mm-hmm.
Can't you?
Well, maybe. I actually think that there's a whole field that we could start to work in women's reproductive health, where we use samples more proximal to fallopian tube tumors, like vaginal lavage.
Okay. And couldn't it be interesting to do that study comparing those fluids from the patients? And
that's been done, and you can separate patients, but remember, those patients have very advanced stage 3 metastatic cancer, so that's not early detection.
Okay.
Yeah.
Okay, thank you.
But that does work, absolutely, and that's the critical data.
Great talk. About the first part of the presentation, you show that Versican helps to migration, invasion. I'm wondering about the first stage of development. I mean, transformation.
Yeah. So we're working on that right now using primary human fallopian tube tissue to study different mechanisms of transformation. As you can imagine, that's a tricky thing to do because ovarian cancer usually occurs in patients, or we detect it in patients that are around 60. There's probably a 7 or 8 year latency period between the stick and that. So, but we are modeling different mechanisms such as P53 mutation, downregulation of PAX2, as well as cancer stem cell expansions.
Maybe some relation with the stick.
Mm-hmm, yeah. Exactly, yeah.
Thank you.
Okay, if there are no other questions, I leave the floor. I thanks again, Joanna, and I leave the floor to the next speaker.